welcome and thank you. Thank you to our conference organizers, Camille Labchuk and Angela Fernandez, for bringing us together for this incredible conference and spending the weekend talking about animals and animal protection and animal issues. Thank you also to the Brooks Institute for their sponsorship. And thank you for being here for this special Scholars Track session with Georgia Mason. Animal welfare and the validity of animal-based biomedical research. My name is Becca Franks. I'm an assistant professor of environmental studies at NYU. And today I have the great honor of introducing Professor Georgia Mason. After this talk, we'll have a moderated Q&A session. So please do note any questions or discussion topics you'd like to explore in more detail during the lecture. Professor Mason is the Colonel K.L. Campbell University Chair in Animal Welfare in the Department of Integrative Biology at the University of Guelph. Trained as a behavioral biologist specializing in animal welfare, her lifelong research interests are in what makes for a good, happy life for animals and how to assess that objectively. Those interests, along with her incredible creativity, rigor, and dedication, have led to publications in some of the most prestigious scientific and multidisciplinary journals in academia, such as Nature, Animal Behavior, Trends in Ecology and Evolution, and the Proceedings of the Royal Society v. Her more than 200 publications have been cited over 15,000 times collectively. The immense contributions of her work have been recognized worldwide for its impact and field-shaping characteristics, and yielded honors including the University's Federation for Animal Welfare Medal and the Creativity Award from the International Society for Applied Ethology in 2022. In granting the award, the ISAE committee writes, quote, "Due to her extensive work, she is one of the most well-known animal welfare scientists around the world, and that beyond that, her creativity is infectious," to which I can personally attest. In addition to her impressive scholarly output, she has a deep commitment to her students and to graduate mentorships. Among all of that, she somehow finds time to direct the Campbell Center for the Study of Animal Welfare at the University of Guelph. And on a personal note, I have to admit that Dr. Mason is one of my personal heroes and has been an inspiration to me and my career, and I really just have the deepest respect for her work and the quality and the clarity of the work that she brings to us. So I think we're all really lucky to have her speak to us today about her latest projects. So I'm delighted to welcome to the podium Professor Mason to deliver her talk.
Thanks, Becca. Well, thanks, that was lovely, and I should say it's mutual, Becca. My talk will be about animals used in research, which are primarily rodents, but it has some more general messages too about the importance of meeting animals' behavioral needs and about the practical value of thinking about animal welfare, even if you're not moved by animal welfare ethically. So I want to start by introducing my co-author. So this is Dr. Jessica Kate, whose work I'm going to talk about. And she defended her PhD about 4 weeks ago and is just now doing a postdoc with me, co-sponsored by the CCAC. And I also want to introduce you to the animals I'm going to talk about. And these are rats and mice. And rats and mice, their lives have evolved to be intimately twined with our own. They typically live in our houses, our factories, our farms, our warehouses. And so we know a lot about their lives, partly because we're often trying to control them as pests. So for example, a mouse who lives in your shed but forages in your house and your garden would have as their home a self-built, well-insulated nest, maybe in a burrow they dug themselves, in which they sleep and they raise their young. And then they'd leave this nest to go on foraging excursions for multiple types of food, for water, for extra nesting material, and also to leave the nest a long way behind to urinate and defecate because they're very hygienic. And so a wild mouse or a rat utilizes a complex 3D range that's at least 1 meter cubed and typically multiple meters cubed in all directions. So they're very good at navigating, remembering each other, copying each other's food choices, and so on. They're cognitively smart little creatures. And mice and rats have been bred in the laboratory for use in research for hundreds of generations, yet despite hundreds of generations of distance from their life in the wild, they still retain motivations to do many natural behaviors. So if people like me run experiments to ask mice and rats what will they want or what will they even pay costs to attain, rats and mice very much still want to use and build nests. They want to sleep within shelters just as they would in the wild. And if they're mice, they're actually cold if they can't do this. They're also still motivated to explore novelty, to access additional space, to have structural complexity and climbing opportunities. They're motivated to run, so they'll make do with running wheels and they'll pay high cost to run in a running wheel. They'll also seek to urinate and defecate as far from the nest as they can. And they also still even want to dig burrows. And if we allow them the opportunities to do all of these things and we ask them, how motivated are you? Mice, for example, in my lab will push weights of 40 grams to get to a cage where they can do all these things, and that's often heavier than themselves, so they're pretty motivated still. So that's the animals, and what I'm going to do today is, in part one, I'm going to present some evidence that the way in which they're typically kept in research labs causes chronic stress because it doesn't allow them to do all these natural behaviors. And then I'm going to provide evidence that this matters not just ethically, but also empirically in terms of the types of research results that we can obtain. And this in turn has implications for validity and replicability of science. And then I'm going to end with maybe an optimistic view of where we might go next. The middle part of the talk is not published yet, and we want to send it somewhere fancy and high impact. So please don't tweet it or X it or whatever it's called these days. Please don't spread it on social media because it's not quite ready for that yet. So I'm going to begin with some published work. And this came out in BMC Biology in December of 2021. And the title has a spoiler. So we found that conventional laboratory housing increases health problems in laboratory rats and mice. And we found this via a technique called systematic review and meta-analysis, where you run an incredibly comprehensive, incredibly transparent search to find all the published science you can, and then run it through some really clear a priori designed filters to collate and then ultimately statistically synthesize all the relevant published literature that you can find. So there's no cherry picking in this work. And it actually began with finding 11,000 hits. That's what Jess had to synthesize to produce this work. So what do I mean by conventional laboratory housing? So if you're familiar with the cages in which rats and mice typically live, they vary around the world, but as a rule, they're kind of shoebox size, and they're typically clear so that we can see the animals, which of course means they can never escape from us. And if animals live in China or the US, all they have to have is sawdust type material on the floor. They will live in something like this for their entire lives. And the vast majority of research animals around the world therefore live like this. So we're talking about hundreds of millions of rodents a year. Now, happily, in Canada and also in Europe, some nesting material and a shelter must be supplied. And these are all evidence-based improvements that were mandated 10 to 15 years ago by the EU and also by the CCAC here. And these definitely make animals' cages better, but they still deny the animals natural behaviors they want to do. They still can't really run greatly. They can't go to the toilet a long way from the nest. They can't burrow. They only get to eat one type of food. They don't get to explore, and so on. And so as a consequence, even in what are globally relatively good cages, even in these cages, we still see many signs of poor welfare. And the most obvious ones are behavioral. So we will see animals indulging in repetitive abnormal behaviors. So here we see a mouse bar mouthing, and she will do this for many hours a day. She's not doing it to shorten her teeth in any kind of adaptive way. She's got the bar tucked behind her incisors and she's just mouthing on it. And experiments show this is derived from attempts to escape. So basically, she's repetitively trying to escape for really hours a day. The mice that don't do that will do things like pluck their own fur out or pluck the fur out of their cage mate. So kind of a somewhat obvious sign that all is not well with these cages, and yet they are the norm. And so in this work, we tested the hypothesis that these cages cause such chronic stress that we'll actually see health problems and maybe even shorten lives. And this idea comes from the vast amounts of human data that show that a stressful life is incredibly physically and mentally unhealthy. So the American Institute of Stress asked rhetorically, can stress kill you? And sadly, the answer is yes, it can. Try not to worry about that, otherwise the worry will kill you. So what we see in stressed humans are people who are under pressure at work or socially isolated or bullied or worried financially, is that the physiological consequences of this cause cellular changes called allostatic load, which make people more vulnerable to health problems, both physical and mental. And what we particularly see is increased vulnerability to what's known as all-cause mortality. So just you're increasingly at risk of dying from just anything, and you're also particularly vulnerable from some key diseases that we know are stress-sensitive. Cardiovascular disease, that won't surprise you. Most of the cancers, that might surprise you. Stroke, you probably know about. And then the mental health problems of anxiety and depression. So if we want to test the hypothesis that these poor-looking housing conditions cause rats and mice stress, they make the testable prediction that compared with better housing conditions, we should see increased all-cause mortality and also greater severity of disease, and particularly the 5 candidate diseases that we focused on here. And we could get data on these diseases because these diseases are deliberately induced in many rodents in order to study them for the sake of human health. So what Jess did was a massive sweep of all the literature to try and find relevant studies. And so we wanted to find experiments in which rats or mice were used in 2 types of housing in parallel. One type had to be conventional or control. Another type had to be what researchers typically call enriched, which means they're enhanced with objects that rats and mice are motivated to interact with. And I've put enriched here in scare quotes for reasons I'll come on to at the end. So this, on top of this, so having these 2 parallel housing conditions, on top, the researchers had to be studying our 5 key stress-sensitive diseases, so cardiovascular disease, cancer, anxiety, depression, and stroke, or they had to be measuring mortality. So here's just one example. So these are images of male mice from a particular strain who are being used in a Chinese study to study anxiety. Half the mice are kept in what they're calling control conditions or conventional conditions on the left, and the other half of the mice are being kept in what they're calling enriched conditions. Anxiety is then induced in various unpleasant ways, and then anxiety is studied in these mice. And Jess found in total 214 studies that were like this. So the other 213, they differ from this. They might use females, they might use rats, they might use different types of strains. They're studying one of the 4 other diseases, but all of them involved a conventional cage and something that we had a priori reason to believe was better. Okay, so from these 214 studies, Jess then extracted data on the severity of symptoms of disease and also on animals' risks of dying. And she turned these into metrics that quantify the impact of housing. And these metrics are known as effect sizes. This might be the most statistical slide. I should have said the cultural norm in science is to have lots of PowerPoint slides while everyone sits there as though they're being sprayed by a fire hose. So that is the cultural norm I bring to you today from my discipline. So effect sizes, you have to turn all this mishmash of data into effect sizes because, as you can imagine, people are measuring different things in different ways. We have data on number of tumours, size of tumours in millimetres, the weight of a stroke lesion, the degree to which an animal is impaired in a task by a stroke, the degree to which it's nervous the degree to which an animal's nervous in a task measured in seconds. And to turn these chalk and cheese metrics into something that we can compare and even combine statistically, we need effect sizes. So effect sizes are standardized measures, all kind of done on a comparable scale. So for death data, we calculated an effect size called a hazard ratio, and a hazard ratio means what's the relative dangerousness of being in two different situations. So a hazard ratio of 1 means two situations are the same. So for us, that would mean that conventional and enriched housing is just equally dangerous, equally safe. And a hazard ratio means one condition is twice as dangerous as the other. So for us, a hazard ratio of 2 would mean that if you're living in conventional housing, your risk of dying at any moment is twice that of an animal living in an enriched cage. For the diseases, we had data on tumour weight and stroke size and so on, as I told you. And we turned these into a metric called a standardised mean difference that looks at the difference in the average value, corrects them by the standard deviation to give a unit-free metric. And the important thing you need to know is a standardised difference of 0 means they're exactly the same. A standardized mean difference of 0.2 conventionally means you've got a small difference. A standardized difference of 0.5 conventionally means you've got a kind of medium difference, and one of 0.8 is deemed large or big. So what did we find? So first of all, we found that compared to enriched housing, the conventional cages did indeed increase mortality. And we found that there were 28 studies that reported deaths in these two different environments, and the hazard ratio was 1.48, which means rodents living in conventional cages at any moment are approximately 1.5 times more likely to die than animals that are living in more complex, preferred, enriched environments. And as a consequence, this meant that the median lifespan was shorter in conventional cages by on average 8.5%. So just for perspective, this is a bit like the difference between median lifespan in Canada compared with, say, Romania or Uruguay. Or within Canada, it's a bit like the difference between being white with a degree and Indigenous with no higher education, no post-secondary education. So that was our data for all-cause mortality, and stress-sensitive diseases, sadly but not surprisingly, showed the same pattern. So we had 72 studies of cancer, and across these we found that the standardized mean difference averaged out at 0.71, which conventionally is deemed medium, and this was highly statistically significant, so the housing systems are differing. And for our 10 studies of cardiovascular disease, we found a standardized difference of 0.72, which is, again, typically deemed medium, and again, this is very significant. For our other 3 diseases, effect sizes were larger still. So we had 56 studies of stroke, and here the standardized mean difference was 0.87. So animals in enriched housing, so I think I forgot to say that the animals in enriched housing are doing better, but I think that's so self-evident. The animals in enriched housing, they're all induced with stroke in exactly the same way by experimenters, but the animals in enriched housing end up less compromised by the stroke and they recover faster. The animals in conventional housing are more debilitated and take longer to recover. In our studies of anxiety, conventionally housed animals were more anxious, and the effect size here was 0.91, which is large. And in studies of depression or changes consistent with depression, the effect size was the enormous 1.24, so animals in conventional housing systems are much more easily induced into states of more severe depression. We found some additional incidental findings, which I want to share with you because they'll be relevant later. So this was sort of beyond the testing of our hypothesis, but they were things that we found pretty eye-opening. So 32% of the 214 studies, or around a third, housed rats and mice on their own, even though these animals are naturally highly social. Studies were male skewed, so over 2/3 only used males, even though by chance, obviously you'd expect researchers to be using, say, 50% males and 50% females. And this reflects a sexist bias in biomedical research that many people have reported on and is obviously still ongoing. In terms of the language people use, we found that absolutely every study treated the conventional cage as a neutral control, and all of them treated the enrichment as though it's a treatment or some optional extra that you're giving to the animal. And the other thing we found is, despite the fact that all these studies are interested in housing, 2/3 of them didn't even describe their control conditions. So we had to kind of infer how these animals lived from local published standards. And then one other incidental finding was that we looked to see whether more enrichments are better, because we wanted to see whether European or Canadian cages really are making a difference. And we couldn't find any direct evidence that Canadian-style conventional cages are healthier for animals than the most bare possible shoebox, but we did find indirect support in that if we quantified the number of resources that are added in an enriched cage compared to a conventional, the more additional types of resources were added, the better the health benefits for the animals in there. See, the effect is pretty damn subtle, as you can see here, but it is highly statistically significant. So what we're finding is, on average, if you add just one resource, that has a health benefit that's quantified as an SMD of 0.7, which is medium, and if you add 5, the benefit is as high as 1.3 on average, which counts as large. It's pretty subtle, but then at least it shows that the more you add, the greater the benefits to animals. This effect is linear. I would expect if we were really reaching optimal welfare, it would asymptote, but I might talk about that later. So our conclusions for animal welfare were that, okay, so our hypothesis sadly was supported, and that conventional cages are so stressful that they compromise animals' health. And this means that preventing animals from doing natural behaviors thus has effects beyond the mere behavioral. They're not just causing abnormal repetitive behaviors, they're actually exacerbating illness and shortening lifespans significantly. And so for rodents in a research context, we think this means that when research is planned and an ethical review, conventional small unstimulating cages should be treated as chronic stress treatments. They shouldn't be treated as the neutral default or the control. You should have to get ethical approval to use them based on an argument that you need to chronically stress your animals for research purposes. And I guess then the other little conclusion is that every single welfare relevant resource you add makes a tiny but important difference to the animals that are housed there. Now, obviously, some of you here are much more interested in wild animals in zoos or in the vast numbers of animals that are farmed for their food. And of course, I think these results are relevant to these animals too. I think that beyond rodents, they suggest it's very likely that if you constrain animals from natural behaviors they want to do, and you keep them in a kind of permanent lockdown situation, that you will see cellular and physiological consequences of the chronic stress that results. And even if these animals don't live long enough to manifest clinical disease, we would probably see markers of allostatic load, immunosuppression, increased inflammation, and this kind of thing. So I think this raises testable hypotheses for other systems in which animals are kept. I think it also raises a red flag about the term enrichment or enriched. We came to believe these terms are just really pernicious because it sounds so kind of frivolous sounding, doesn't it? I'm going to enrich my animals. It makes it sound like you're doing something really lovely and that you're giving them chocolate-covered strawberries. And obviously you're really not, actually. What we're talking about is remediating chronically impoverished conditions that are actually not meeting animals' basic needs. So from now on, I'm not going to use the term enrichment or enriched. I'm going to just talk about resources. So I'm going to say the more resources we give animals, the better their welfare is. Okay, so in the second part of the talk, which is the unpublished work we're still finessing right now, I'm going to give you some evidence that these impacts on rodent biology do actually have some implications for the validity and replicability of the research results that are generated in biomedical research. And we did this because we kind of realized that animal welfare arguments are often just not really enough to affect change. So after the BMC biology paper came out, we kind of sat back and waited for media attention, and there kind of wasn't that much. We got a Globe and Mail article, which was great. We got coverage in Israel in a broadsheet there. That was great, but really nothing else. And the lay non-science journalists just couldn't get beyond rats and mice being vermin. And the science journalists just said, we know all this already, which left us kind of stuck. And we would say things like, but mice, they're so cute and adorable and they're intelligent. Mice really like having their faces rubbed, like you can see here. And rats are almost unbearably cute. They can count to 10. If you feed them a Cheerio, they look like they're eating a bagel. Like, how can you not be moved by these animals? And it didn't work at all. So we just realized the sad truth is that rodent well-being just isn't a concern for many people, and that if we want to affect change, we need to talk about the benefits to humans. So we sat on our love for these animals, and we decided to take that approach. Okay, so that conventional housing reduces welfare is old news, and it had obviously been not enough to affect change. Why hadn't it really affected radical change in the research world? Well, one obstacle, aside from public indifference, is that, as is common for animal welfare issues, change is very expensive. And so if all rodents were given larger cages full of resources, this would cost a fortune, and who's going to fund it? So that's always a major obstacle. So money is one issue. But a second obstacle is that researchers are often very concerned that they would lose results that they're used to finding in conventional cages. So they might work with a particular model, and they know that, say, a particular drug always works on that disease model, and they're very nervous about change because that would compromise the rate at which they can produce publications, which is the thing that academics like me are rewarded for. So researchers are just very scared that they will lose results if they change their housing conditions. And in some ways, this is a fairly plausible concern because we've shown that conventional housing has a whopping effect on biology. And there's also some odd kind of examples out there where changing housing does indeed change what you might learn. So one example is that, for example, there's evidence that resveratrol, which is a compound found in grape skin, has antioxidant effects which can be really healthy. And we know this from conventionally housed rodents. So any of you who think red wine is good for you, this is where you've got this idea from. But it turns out if you replicate this work in well-resourced animals who aren't under stress and therefore don't really need extra antioxidants, you no longer see this result. So red wine may be good for you if you're miserable, but if you're happy, it probably doesn't make that much of a difference. And so examples like this suggest, right, your results, what you conclude from a research experiment can perhaps change if you change how animals are kept. So this was the hypothesis that we tested next, that improving rodent housing alters their biology so much that research conclusions generated actually change. And this hypothesis makes two testable predictions. One is the obvious one, that the effects of drugs that you might be exploring, or genetic knockouts, or diets, or other disease modifiers that researchers are investigating to understand their impacts for humans, the effects of these various disease modifiers should differ between conventional and well-resourced rodents. Sorry, WR is well-resourced. So well-resourced and conventionally housed rodents should generate different results in a way that would actually change what you conclude. That's one prediction. And the other is that these sort of disruptive effects will be most manifest when improved housing really has a big change on measured aspects of disease. Because we'd expect that if housing is having a whopping effect on disease, it's not just changing the animal quantitatively, it's changing it qualitatively such that drugs and so on start to work in an actual different way. Okay, so to test this hypothesis, Well, I want to first go over these predictions in a bit more detail so that you can see what they look like sort of statistically, so that you'll be convinced by the data when I actually show you them. So these are the predictions again. So there are actually 4 possible ways in which research results can change if you change an animal's housing. And one of them is a bit of a red herring. And people have sometimes run off with this and said, see, it's important. But actually, the first way I'm going to talk about actually isn't important, and doesn't support our hypothesis. So that's the example I've shown here, where if you change animals' housing, you change the average values of everything you measure, but the actual conclusions you draw are identical. So let me talk through what I mean by this. So here we have some data. I've completely lost my cursor. My cursor is over there somewhere. Oh, here it is. Okay, so here we have some data from 2 populations, a conventionally housed selection of animals living in shoeboxes and a well-resourced population of animals who are kept in better conditions. And on the y-axis, we have a measure of disease severity or disease morbidity. How severely are they getting ill when they're induced with cancer or stroke or whatever is happening in that lab? And they're being given a disease modifier, which is a drug shown by the grey bar. So what you can see here is that the conventionally housed animals are becoming more severely ill, the well-resourced animals less so. And this is true whether they're not given the drug, the white bars, or whether they are given the drug, the grey bars. But in both of these housing conditions, the drug is really effective. It's reducing the disease severity by half, and you have a statistically significant effect of the drug shown with a red asterisk. So in this condition, which I'm going to call pattern A, housing is causing a change, but it's not one that would alter your research conclusions, because both these groups of researchers are going to conclude that the drug is good. What's more important are cases where, for example, the drug proves to be much more impactful, or perhaps only impactful, if you keep animals in conventional conditions. And that's what's shown here in what I'm calling pattern A. So here, the drugged animals are in black, the control animals are white. We have conventional and well-resourced again. And we can see that if you're a conventionally housed rodent, the drug is really effective. And if you're well-resourced, the drug doesn't work at all. And this is like the resveratrol example I just gave you earlier. So that's one case where your conclusions would change with housing. And of course, you might see the opposite. It might be the case that you try a drug in conventional housing conditions and you think it doesn't work and that it's a bust. But actually, if only you'd housed your animals better, you would discover that the drug is highly effective. And there are some examples out there in the literature as well. So this pattern C is the second pattern that would support our hypothesis. And there's even potentially the possibility that what you find completely flips in direction with housing. So in this example, pattern D, the drug is beneficial to conventionally housed animals, but actually harmful to well-resourced animals. So that's one way in which you could get a complete flip. And of course, you could see the other one, that the drug is great in well-resourced animals, but somehow bizarrely harmful in conventionally housed. And so this pattern D is another one that would support our hypothesis. So our hypothesis predicts that we should see patterns B, C, and D, and technically these are known as statistical interactions between housing and a disease modifier like a drug. And an interactive effect just means your effect is nuanced. The effect you find of a drug is contingent on other things like housing or well-being. Okay, so that's a summary. So our hypothesis predicts that we'll find B, C, or D patterns, and if we find pattern A, we're not going to be distracted by it because that's a red herring. The second prediction is that if it's large underlying changes in biology caused by housing that are having this disruptive effect, then basically the larger the housing effect on disease severity, which is what we found in the first study, the greater should be the interactive effects between housing and drugs. In other words, the more your housing benefits rodent well-being and makes them more healthy, the more likely it should be that the drug effects and what have you that are that people are exploring are qualitatively completely different, the more likely it is that you'll find completely different conclusions. So here we've got the effect size of housing on disease severity, which are all the metrics I told you of already. And on the y-axis is the new things we're going to calculate, an effect size that summarizes how different research conclusions are in different housing conditions. Okay, so I'm going to come back to this graph because some of you are looking a bit googly-eyed, but don't panic, we're going to come back to it. All right, so we tested this hypothesis once again using systematic review and meta-analysis. So Jess updated her giant trove of gleaned papers, but this time she sifted through them to find studies that were not only looking at disease and not only looking at two housing conditions, but also in both housing conditions, they had a disease modifier and a control, maybe a drug and a placebo, maybe a Western diet and a healthy diet, maybe they were looking at males and females. They're always looking at something else that's cross-factored or orthogonal to the two housing conditions. That just summarizes that really, and from this, Jess extracted means and standard deviations again to calculate these standardized effect sizes, to calculate an effect size that quantifies how different is the effect of a disease modifier if you look at two different housing conditions. And essentially, this is an effect size that assesses the difference of a difference. So what she would do is calculate the effect sizes of a drug in conventional housing, the effect size of a drug in well-resourced, and then assess whether these two effect sizes are different. If they're not different, they're both equally effective in the two conditions or equally ineffective, the difference of the difference will be 0, which means the same thing's happening in both. If we find a standardized mean difference of 0.2, again, this means, there's a bit of a difference. There's a small difference between the two housing conditions. 0.5 is medium, and 0.8 means large. There's a big difference in your research conclusions between your housing conditions. So this is what her data set looked like. So she managed to find 70 articles containing 97 individual studies that met our criteria. They looked at two types of housing and then a disease modifier and some kind of control. Very similar studies that were, for example, looking at the same drug and its impact on the same aspect of disease. She pooled for technical reasons, and this led us to, I say us, Jess did all the work, this led us, or Jess, to be able to calculate 64 effect sizes that summarized how different research conclusions are between different types of housing. And these 64 effect sizes spanned all 5 of our diseases. So we had 26 on cancer, we had some on stroke, some on depression, some on anxiety, and a handful on cardiovascular disease. The disease modifiers themselves also varied. So nearly 2/3 of them were potential therapies, as you'd imagine, because that's what people are always striving for. So 60% were drugs that were intended to alleviate the disease. But around 1/3 were modifiers being used to probe disease mechanism, and sometimes they might even deliberately make diseases worse. So for example, like giving animals Western diets to make their cardiovascular disease worse. And then 5% of the disease modifiers were demographic things like how does sex affect disease severity? How does age? How does your genetic background? Okay, so this is what we found. So this way of showing data is what's known as a forest plot, and each black square summarizes the standardized mean difference or the effect size from a particular disease modifier, let's say a particular drug, and the little whiskers above and below it just show the 95 confidence intervals. So this basically shows this is our estimate and this is the kind of fuzzy area around each estimate. So each one of these is an effect size reflecting a particular disease modifier such as a drug. Now, if the difference is 0, that means both housing conditions are generating the same conclusions. But if they're above 1, they're enormous. And what you can see here is sometimes we're getting enormous effect sizes that show you that there's enormous differences in research conclusions between housing conditions. Okay, so the handful I've shown here are 19 disease modifiers that act in the same direction in both housing conditions, but they did so much more effectively if rodents were conventionally housed. So that's what's happening here. The drug, for example, is much more impactful in poorly housed rodents than well-housed rodents. And for 8 of these, this difference was statistically significant. So that's what's important. For 8 of these, we've got a statistically significant difference in research conclusions based on housing condition. So the pattern I'm showing, and that's this 8. So the pattern I'm showing here is like pattern B. That's like the resveratrol or grape skin example. So here, patterns, drugs that, for example, look great in conventionally housed animals are inadequate or even failing if animals are well-resourced. In other cases, we found that, again, the disease modifiers were working in the same direction, but now they were much more effective in well-resourced animals than in conventionally housed animals. So if you were only working with shoeboxed housed animals, you'd think your drug didn't work, house them well, and you suddenly find it does work. And for 8 of these 30 cases, the difference between housing conditions was statistically significant. So that's the 8 shown here. And these 8 are following pattern C. So here, drugs that look like they're not working in shoeboxes are looking like they're actually working if you give animals well-resourced conditions. And extraordinarily, we found quite a few studies in which the research results actually flip direction if you house your animals differently. So for 15 disease modifiers, the effects of them pushed animals in different directions based on their housing, and for 7 of these 15, this flip in direction was statistically significant. So that's the 7 shown here, and here you can see they have some really quite large effect sizes. And so for these 7 studies, this is like pattern D. So we're actually drawing completely different conclusions based on a drug according to how we house our rodents. Okay, so this is the overall summary. So in about a third of our cases, we showed that improving housing makes what you discover in conventionally housed, poorly housed animals non-replicable. You can't extrapolate from conventional housed animals to animals with better welfare. So our first prediction is met at least for around a third of our studies. Our second prediction was that the more the housing affects the disease, the more likely it is that you're going to get these changes in magnitude or even changes in direction of how, for example, a drug works. In other words, I predicted that the housing effect on disease severity, our measure from study 1, would predict our new interaction effect sizes that quantify how different our research conclusions based on housing. And that's exactly what we found. So what this shows is basically it really is driven by the underlying changes in an animal that aren't just quantitative, they're clearly qualitative and really quite disruptive. Okay, so does housing quality change research results? Should researchers be worried about improving their housing? So most of the time, no, especially if you're not really improving housing that well, so you're not really making that much of a difference to the underlying disease. So 64% of our cases didn't show any of these significant interactions, which are the important thing. But in 36% of our studies, the modifier effects, the drugs and so on that were under investigation, did significantly interact with housing such that the conclusions researchers draw would differ according to house their rodents. And these interactive effects are particularly marked when the housing differences are having an enormous effect on disease. So for example, if you're comparing really good cages with really terrible cages, you're going to be particularly likely to then run away with completely different research conclusions. Okay, so we think this has considerable implications for the replicability and external validity of biomedical research. And now I want to show you why this is important, because these are 2 enormous underlying problems in biomedical research. And I know some of you will know this, but those of you who don't know this, be prepared to be pretty shocked. So poor replication and poor external validity are 2 widespread enormously entrenched and enormously expensive problems that really bedevil biomedical research. So let me explain. So first of all, replicability means the ability of researchers to find the same results when they repeat their own or someone else's experiments. So what this means is if you read something in a journal and you decide to run the same study, you would hope to find the same results because science is kind of built on the assumption that what you read is a truth that transcends just that one individual lab that did the work. And yet meta-analyses of biomedical research results show that in 51 to 89% of cases, preclinical research involving animals cannot be replicated by other people. So this is an enormous problem for researchers, it's an enormous problem for anyone reading journal articles, and it also means that even if we're charitable, half of the funds spent on biomedical research is being spent on non-replicable kind of ephemera, and half of the hundreds of millions of animals being used are being used in experiments which are, again, almost like the results are almost like will-o'-the-wisps. So we wonder whether, and obviously this costs money and it wastes animals' lives, and so we wonder whether understanding and recording rodent housing would improve replicability. As we've seen, typical rodent housing varies between countries. It also varies between labs because some people are always cutting corners, other people are really valiantly going beyond the norms to improve their animals' lives. And yet, despite its a clear effect on research results, most people aren't reporting how their animals live. They're not required to by guidelines like the ARRIVE guidelines, which are reporting criteria for biomedical research. This just says, if you want to report housing, that would be kind of nice, but the ARRIVE guidelines don't require it. And as you saw, even in our studies done by people interested in housing, two-thirds of them, no wait, I can't remember if it's two-thirds, anyway, a hell of a lot of them aren't even describing how their animals are kept, which obviously means how can you replicate a study if you don't know how the animals lived, when how the animals lived is incredibly important for your research findings. So that's one way in which our results have practical relevance. But of course, the other issue is if research conclusions can vary according to house your animals, then which type of housing is most relevant to most humans who ultimately people want this work to apply to? And in a way, this is kind of the question, if you are a rat or a mouse, what kind of a rat or mouse are you? What kind of rat or mouse would most represent you if you were being modeled by a rodent in a research lab? So what kind of rodent are you? Oh, that's got a bit squashed. So this is actually a rat called Georgia, who's owned by a friend of mine. And like me, she's female, she's middle-aged, she's never had offspring, she's maybe a little bit scrawny, but she's also well-resourced with good social relationships, making her pretty happy. And so I would say that Georgia the rat is a fairly good model for Georgia the person. And both me and Georgia the rat are not like most research rodents because we're not isolated, unstimulated, chronically stressed, overweight, or male. And why might this matter? Because most data that are allegedly being applied to all of you come from overweight, stressed, isolated male rodents who live these very peculiar lives. And this in turn matters because even when biomedical research can be replicated and a pharmaceutical company goes, right, this is great, we're onto something, let's run it out into clinical trials, these clinical trials almost always fail because the animal data almost always fail to translate to humans. And this is known as poor external validity or poor translatability. And so the data are pretty shocking. So even when we look at replicable research, which is under half, of those promising results, only 9 to 14%, so let's say around 10%, successfully translate to humans when they're run through clinical trials. And once again, this is an incredibly financially expensive problem. Failed clinical trials worldwide probably cost about $90 billion a year, and it's just a problem that no one wants. And of course it means that yet more of the animals' lives that were involved in producing this work are going nowhere because the animal data are not producing anything relevant that can actually solve human health problems. And we wondered that one issue is that there are false leads being generated by conventionally housed animals. Something looks like it works in a really weird stressed animal, and yet when you apply it to the normal human population, it stops working. So a pattern like pattern B in our data is one where, yes, it only works if you really look at really stressed people. It doesn't look at normal people. Biomedical research is prone to false negatives as well, rather than just false leads. So we know that there's all kinds of things that we use happily, like aspirin and tamoxifen and penicillin, that had they been run through today's research and biomedical screening procedures, they would have failed. Yet thank goodness they didn't go through those, because we've got to benefit from them. It's hard for us to quantify these false misses, but we wonder whether our pattern C, where things only work if you test them in enriched animals, explain some of the false negatives that bedevil translational research. So we think improved translation would be a benefit of understanding and improving rodent well-being. We think the status quo may be extremely relevant for lonely, chronically stressed, overweight, institutionalized male humans, but perhaps not for the rest of us. And that if we want to model, and of course we may want to move away from animal research, I know that's what many of you are thinking. If we want to move, if we're going to stick to animal research, we should be modeling a more diverse array of lived experiences by housing animals, not just in terrible conditions to model humans who have terrible lives, but in good conditions to model many people in the Western world, and maybe even phenomenal conditions to model those of us who are lucky enough to have very good lives. So improved rodent housing could improve their health and welfare, which obviously is the most important thing for most of us. But if that doesn't get traction, a kind of practical benefit is we think it could improve replicability and improve translatability, which would better find effective disease cures for humans and save tens of billions of dollars, which could be better spent on other things. Now, I'd love to tell you that the whole answer to the problem is animal welfare, but of course it's not that simple. Otherwise, someone would have found that out already. And the truth of the issue and the truth of these problems in biomedical research is that the poor replicability that we see here and the poor translatability actually reflect a complex array of other problems. It's multifactorial, and the whole issue is really what the wonderful Amy Schumer would term a hot mess. And the reasons for these hot messes, they don't just apply to biomedical research, they really apply to a lot of academia. So in the models we use may be wonderful in the lab, but they may not apply to real patients. Even if we're using happy enriched rodents, it still may be inadequate to use animals. There are publication biases across the literature, because if you fail to find something, it's really hard to get that published. So null results don't make it out into the public world. All you see in journals is statistically significant results, which gives you a really weird skew of the status quo. And there are biases within studies that reduce their internal validity, or literally their truthfulness, from people doing things like run multiple statistical tests till they find something, wringing their data in all kinds of creative ways to find what academics like me need, which is a statistically significant result that we can actually publish. And so the problem really is that across research, as the editor of The Lancet said a few years ago, no one is incentivized to be right. Instead, we're incentivized to be productive. And I think we all know that you kind of get what you incentivize, even if that's kind of crazy. So that's a bit gloomy. So I want to end with some what I think might happen next, and also some reasonably optimistic thoughts, particularly about the situation in Canada. So I do think that science in general has to slow down and to increase how much rigor is valued over how much the rate of productivity is valued. And we also need to use more tools like registered reports, which is where you have your good idea, you describe your methodology, that goes out for peer review, and you then get, if it passes, a guaranteed publication, even if you get null results. So registered reports, the increasing use of those, could solve a lot of the weird, perverse incentives that underlie a lot of these problems. the health and well-being of research animals should be assessed and reported and justified when choosing to work with animals in a particular way. And this means described and justified when people get grants, when their work goes through ethical review, and when they send their work out for publication and it goes through peer review. And I think in Canada, the CCAC needs to be particularly congratulated on moves. Now, the CCAC is incredibly kind of powerless, but they really are trying to do their best to affect change. So the CCAC is now requiring that research animals' home cage welfare is assessed. Now, currently, I would say this isn't really happening, even though it's a requirement. And currently, I would say it's seen as a kind of onerous obligation, you know, it's top-down control, blah, blah, blah. But actually, if it was implemented and actually started being seen as part of science, then understanding animals' welfare and how it affects research results could start becoming an integrated, holistic one. So I think if understanding animal welfare becomes part of science and not just part of regulatory control, that is a way forward. And the CCAC, as I mentioned, is sponsoring Jess now to look at the effects of social isolation on rodent health and welfare, but also again on the replicability of research results to see if we can make some impact there as well. All right, so that was a lot. You were very brave. I want to end with some thanks. So thanks first and foremost to Dr. Kate, who did this work, my lab, NSERC and U4 for funding it, the Campbell Centre for the Study of Animal Welfare, and also to MITAX and the CCAC for funding her work moving forward. And I'm happy to take questions.
So I think just to reflect a little bit, I have 4 questions that I'd like to ask and then open it up to the audience as well for further questions. But you know, where we ended here, there's this replication crisis, there's a translatability crisis, there's all this money that's being wasted, lives are being wasted, and we're probably even missing valuable therapeutics. So it's a total disaster, ethically, financially, as your work is touching on epistemologically as well. We're just not understanding the world anymore. So I think there's just really huge, profound implications for policy and inquiry going forward. And, you know, at the risk of making you be reactively humble, your work does show that animal welfare is playing some substantial role in that, and that a lack of attention in particular to animal welfare is playing a substantial role if it can't account for the entire thing. But I want to pull out something you mentioned earlier in the talk just as sort of a potential entry point for this, is that this idea that we have evidence now across multiple studies, as is shown in a meta-analysis, that these conventionally housed systems are actually chronic stress manipulations, and that if you were going to chronically stress an animal and pass an ethical review board, you'd have to justify it, that it's necessary for the research. And so I wonder if you can just talk a little bit more about that reframe and how that might play out.
Yeah, sure. I think, do I have to borrow this? Oh, no, I'm wearing this. So currently, a researcher who wanted to study chronic stress, which they might want to do because chronic stress plays a role in so many illnesses and also things like drug addiction, they would have their conventionally housed animals and then they would subject half of them to additional stressors, which would be things like food deprivation and electric shock and isolation. And they would only do that if they got ethical approval. But I think the idea of where those comparators are on the axis of misery needs to be shunted up. So instead of saying, right, you're comparing miserable animals with even worse off animals, you move everyone up so that the conventionally housed animals are now the miserable group, and you compare them with animals living better lives. You get the same phenotypic effects, there's the same underlying logic that people with better lives are less likely to become addicts or depressed or what have you, and yet all the welfare is better. Some animals are still relatively stressed, because if you want to model stress, you have to model animals. And in today's society, that's deemed legitimate, rightly or wrongly. But you could do it in a better, more ethical way than the status quo. Yeah, so that's what I think it would look like.
Great, Thank you. Then in terms of the extension, so this work, the meta-analysis itself, of course, was with laboratory rodents. I'm assuming just mice and rats,
Yep.
not like hamsters or. So you touched on briefly how this could extend into other areas of animal use, and I personally, as some of you might know, have an interest in fish, but obviously fish are of interest to many people, not just me. They're increasingly used in laboratory settings in particular, and again, going through this same pathway of justifying their use because they actually are so similar to us and being able to develop depression and get addicted to drugs and having similar disease progressions. So they're increasingly used as replacement, as less ethically problematical, even though they're so similar to us. Anyways, but so there's fish, the aquaculture industry is expanding. And then, of course, also, even furtherly distant relatives of ours, like invertebrates, we're starting to discover not just in the arthropods like bees, but also in mollusks. We have octopuses demonstrating so many different capabilities, but even in their relatives, mussels, clams, etc., can show signs of stress. So my question is, when we're thinking about extending the implications of this work beyond the laboratory, beyond rodents, do you have any thoughts about how to do that, when to do that, and then also I think crucially where the burden of proof should be? Should we prove that there's a problem with these incredibly sterile, barren, monotonous environments, or should you have to prove that there's some added value of providing more resources to animals?
So I'm going to pass that out into two things. So there's research animals beyond rodents, and then non-research systems. So I would say I think our results have relevance to, say, farm animal systems, but I think I'm looking at Lynn right now, it's much more difficult to affect change because there's two enormous conflicts of interest. The interest of industry to make money and the interest of consumers to save money. And so the consumer buying cheap products and the farmers trying to make money push against animal welfare in a way that makes that, it's not impossible to change, but really hard. What's good about the problem for research animals is that no one wants this problem. It's currently such a waste of money in animals' lives that no one wants it. And I'm hoping that will be a goad for change. Do our results have implications beyond rats and mice? I mean, I'm sure, because we see the same types of compromised welfare and the same manifestations of welfare problems. If you go to, the worst North American rhesus macaque facilities, animals are doing so much abnormal behavior and plucking their hair out, and we know that this is a product of stress. What on earth do we think we're modeling there? We're modeling Romanian orphans, we're modeling people that have had terrible lives. Of course, people like that need medical help, but are we doing that deliberately? No, we're doing it by accident. And you mentioned fish, those zebrafish are commonly used. If we can't get people to care about research rats and mice, they're really not going to care about zebrafish. I'm sorry, they barely have faces. They're not used. But the same thing applies, that if you house them better, they live longer, actually. And if you house them better, they become more intelligent because their cognitive systems are more developed. So I think their value in use would improve if their welfare was improved. And you and I would care about their welfare, and other people would care about their usefulness. And I'm hoping that hand-in-hand thing will eventually cause change.
Great, thank you. Okay, so transitioning to a little bit talking about the changes that are required. So I'm curious to hear more about what sort of structural changes in science could happen to sort of jolt us out of this. You mentioned changes in publication bias, but I think there's also just inherent biases in science about having, as we talked about earlier this morning, about having this fear of this notion of anthropomorphizing. But really what we mean by anthropomorphizing is not biologically anthropomorphizing animals, which is, I think, at the root problem here, is that we're mistaking animal biology for human biology, or rat biology for human biology, which is a form of anthropomorphism. But instead, using this word anthropomorphism to talk about anything to do with their consciousness or their mind or the subjective state, et cetera, et cetera. So beyond sort of the publication structural issues, there's sort of bigger social norms in science. around that could also be driving this inability to see the welfare of the animals as being more than just like taking good care of a Bunsen burner in your laboratory?
Well, I think there's a couple of possible routes in. One is I just think we need better, more inspiring educational tools as to what rats and mice and zebrafish are capable of and do in the wild, so that they are manifestly less like furry test tubes and more like creatures who constantly make decisions and are beautifully adapted to their environments and have cognitive abilities that are surprising. I mean, a rat, if you handle a rat will remember you, the individual handler, for over a year. Rats have dreams. Rats, there's great evidence that rats can count to 10. They have episodic memory. I think most biomedical researchers don't know that and would be interested in it because they're using these animals to model humans. And these genuinely human-like traits, I think they'd find them inspiring. The other is a very practical change that I would suggest. So currently, rats and mice are nocturnal, and most biomedical facilities have lighting systems arranged for us, not surprisingly. So the lights are on in our day and they're off at the night. So when you go and, if you visit rats and mice, they're just sleeping because for them it's when they sleep. So a sleeping animal kind of looks fine. So if you go into the average biomedical facility, you're just seeing loads of rats and mice fast asleep. When they wake up, when the lights go off, that's when you see that they're just spending hours a day doing stuff that's really crazy. And I think most biomedical researchers would be a bit shocked to realize that the mice their data come from a mouse who's doing kind of wall of death backflips for 3 or 4 hours a day, because they literally don't see that. And so one thing we've started suggesting is just adjust your lighting regime a bit. Not completely, because it is hard work groveling around in the dark or under red light. Just adjust it a bit so you see an hour or two of their actual active behaviors, and then you can see how weird they are. Or some of the animals are just sitting there, staring into the middle distance, not doing anything for 8 hours a day when they should be busy doing stuff. We think they're probably depressed. Well, so I think just actually watching your animals in situ would make a difference, and just honestly tweaking the lighting regime would help a bit. Yeah.
That's great. It's very practical and doable, it seems.
Yeah.
Modification. All right, I have one more question, and then we'd like to open it up, and I do have many other questions as well, so don't worry. I will fill the silence if there is any, but I do hope we have some good questions from the audience. Considering the ethical, financial, epistemological disaster, the hot mess, I do think that, like, at least talking about this idea of just actually stopping this madness, what if we started considering heading in that direction? I actually have a question about what would happen to the scientists, all these people who would suddenly lose their jobs or their livelihoods. So in agriculture, we're starting to talk about a just transition for farmers. Could we have a just transition for scientists and figure out new things that they could contribute to society? And if so, what would that be? And then also, I think you have to, the ethical dissonance that a lot of researchers, biomedical researchers experience, because this is not coming from a place of sadism, of course, of like, harming animals in this way, but instead coming from an ethical, a weighing of thinking that, like, oh, this is so important. Now we're discovering that actually a lot of the data is not even translating to humans. So I think there's like some trauma also to be accounted for too. So I'd love to hear your thoughts about that.
Yeah, well, that's one of the things that gives me hope. I mean, most people are horrified to know that much of their data is going to be irrelevant. It's not why people go into this. And yes, but it is very difficult to change what they do for a living. I mean, I am an animal researcher looking to get out of animal research because I'm finding it increasingly distasteful. I'm having to retrain myself to do meta-analyses of existing data. And it's tough. And it's tough for people to move away to non-animal methods because it requires a whole load of skill sets that they don't have. But of course, moving away from animal research would stop some of the ethical cost of this. It won't solve the problems that non-animal research is still full of non-replicable results and non-translatable results and people wringing their data out until they get something significant so they can publish it in a fancy journal. I mean, the perverse incentives of science also need to be dealt with. And I kind of think, I know one welfare is a bit woo-woo, but I think there's a one welfare issue that, you know, academia is a tough place to be because we're in a constant arms race with each other. We're always competing for the same scholarships, grants, promotions. And so we're always trying to out-publish each other, which means it escalates in a crazy way. And it's like we need a reset. There should be a slow science movement. I speak as someone who works 6 days a week. I would like a slow science movement, but I'm trapped on the treadmill along with everyone else.
Okay, great. Thank you so much. So are there any questions in the audience? Yes, I see one over here.
Yeah, thank you so much. This is really fascinating, and I'm so glad you're doing this work. And I have a lot of questions for you, but I'll start with two, which are both, I guess, I'd be curious if they're kind of, I guess, extending this a bit, and I would just be curious to hear your thoughts on these. So the first is that there have been a couple of calls for like so-called, I guess, like free-range lab animal schemes. There was a great article, surprisingly great article in the New Yorker about this like a while ago, where basically I think the argument was to keep lab animals in not in the lab, but rather, you know, you might have like kind of a field of grass where your like rodents run around, which obviously creates like tons of problems with you know, causation and like actually getting like valid data about the effects of whatever you're trying to test. But I was wondering if you had any thoughts on like circumstances under which that might work or that might be like scientifically useful. And the second thing is picking up on something you said a few moments ago, which is, you know, a lot of the stresses that these like lab rats and mice are forced to undergo are quite extreme. So like electric shocks, simulated drowning, deprivation of food and sleep. Like if you did that to humans, that would be basically like torture. And I was wondering if you had any thoughts on like to the extent to which we continue doing experiments on animals, if there are like better ways to induce stress in them. I mean, obviously like any kind of induction of stress is like not great, but maybe there are like less torturous ways to do it that are also more scientifically valid.
Yeah. I mean, I'll start with the last one. So yeah, so I think to use those techniques to inflict stress on an animal who's poorly housed and who may well be socially isolated is so gratuitously stressful, we don't need to do it. You're absolutely right. We're modeling something very extreme if we were to turn that into a human situation. And if you want to chronically stress a rodent, all you have to do is keep them in the status quo and have a more nicely housed control group. So I really think the arguments for stressful procedures should start to be criticized as superfluous. We just don't need them because current housing is a stressor. That will take a while to take foothold, I'm sure, but that's what I would argue, that we shouldn't be using things like chronic unpredictable stress to induce depression when their housing systems are doing that already. I guess that's what I'd argue. Then you argued about keeping rodents in more free-range systems if we're going to use them, and I think that would be great because then you've got animals that are more behaviorally and cognitively normal and their stress levels are more normal. And they're also kind of dirtier. That's part of this other argument, that that the artificial cleanliness of your average research lab also does things to the microbiome that make them really peculiar. And that if you want an animal to really model a real animal and also a real person, they need to live in a grubbier environment. So I kind of think that would be great. What has to happen is that researchers have to be bold enough to want to try that. They have to be supported by their universities in terms of the facilities that they can create and that can be supported, and that funders then have to pay for that. So that's what has to happen. And, you know, money is often the incentive and the obstacle. Yeah.
Okay, great. I see a hand in the back.
I'm coming. Thanks so much. That was super compelling and interesting. And yeah, just one thing that I think actually further corroborates and strengthens your argument is kind of even in your first case scenario where you have kind of no statistical interaction on the difference scale, but you do have this shift in levels. It's interesting that when you have that shift in levels, it means that at least one of the difference or the ratio scale effect measure will change. And so even if it's not changing the difference that they estimate, it's going to affect the ratio. And so it also is really interesting because you bring up these points around publication bias and kind of cherry picking and manipulation of analyses. And so, I mean, that's a case where if a study is reporting on both the ratio scale, so the risk ratio, hazard ratio, and difference scale, as they often will, one of those, at least one of those, is going to be affected by housing, even if it's not the difference scale. So, then which one gets reported in the media? Probably whichever one mattered more. So, I mean, it's just kind of interesting that even in that case number one, it probably still matters that there's this effect. So, thank you again.
Thank you. Thank you very much for your presentation and your research orientation. I appreciate that. My question is about COVID and the impact it may have had on animal research. The CCEAC does their very best within limitations. I really don't know what happened with COVID. Were macaques brought in from all over the world to Canada and everywhere else? Were some of the regulations thrown out the window? And I can just imagine that the stress of all the facilities would have increased. And then we ask about validity about that data. Thank you.
That's a great question, and I can't answer all of it. But I would say no. I mean, certainly in Canada, people wouldn't suddenly start bending the rules because we're in a pandemic. The rules apply, and you still have to get ethical approval and so on. So I don't think anything that sinister would happen, at least here. I do know that I've got some colleagues in Edinburgh who monitor research quality and the kind of potential for bias within a study in terms of an individual study following best practices. Are they randomizing animals between their treatment groups, or are they taking the first 20 animals out the box and putting them in one and the second 20 in there? Are they blind when they run an experiment? So do they know which treatment group is which, or do they not know? If you know, there's evidence that can bias you to find the results you want. And all of these potentially bias-introducing practices were logged in COVID studies, and they were appalling. I mean, they were almost worse. I think people did cut corners there. So they probably couldn't cut ethical corners because there were procedures. But once you're in the privacy of your own lab, well, I've got to solve a pandemic, and then people are doing terrible science in the name of solving a terrible problem. It's kind of insane. And there are some issues there around just the statistical education and experimental design education for biomedical researchers and local cultures within labs. Students are often quite well trained in this, and then they'll join a lab and they'll just have it beaten out of them. We might, you might have learned that in class, but in the real world, we don't do that. So that kind of thing is pretty shocking. Yeah. Does that answer your question? Yeah.
Thanks so much for that very interesting talk. And I wanted to ask you about whether you think we can use your findings to draw any conclusions about cognition and the kind of cognition studies that are done in different labs. And I'm thinking about things like chimpanzees, where in some labs, researchers are saying, oh, there's no cooperation, chimpanzees are just competitive. And in other labs, in Japan, the chimpanzees are cooperative and show different sorts of behaviors and pass different kind of cognitive tests. So is this enough for us to draw conclusions about these, or could we do meta-analysis studies, Or are there just not enough studies about ape cognition?
Yeah, what a great idea. And I think your sub-question is totally right. So commonly what happens is people just buy their animals from Charles River or some company like that, house them conventionally because, hey, that's the convention, run cognitive tests, and then might find that a certain type of animal can't do something, or like you say, they're not cooperative, or they, mice don't have episodic memory, or rats can't do so and so. But of course, what you're looking at is animals who've been taken from their mothers prematurely, housed in impoverished conditions all their lives, and in conditions which make animals more aggressive. Actually, this is the other thing we know from welfare research. So that's as though you're collecting data from the most impoverished types of people and extrapolating from them to humankind, and it really makes no sense. Yes, it really makes no sense. It'd be great to do a meta-analysis, actually. But yes, of course, if you want to say something about the abilities of rats, you have to allow the rat to meet their full potential before you then test the rats. Otherwise, it's just not fair. Yeah. And it's like some of the old cognitive stuff where rats were set visual tasks. Oh, look at that, rats are rubbish at visual tasks. Yeah, it's because their eyesight isn't that good. If you do a cognitive task, they show you they have the intelligence of a 2-year-old human. It's just, you've got to make the situation allow the animal to flourish, and then I think we'd be amazed. Yep.
Yeah, it's a great model for expanding to lots of different areas, I think, this comparative work. We have a question at the front here and then at the back.
First off, I found that so enlightening. It is so great for so many arguments that, I think we're always confronted with, that we need that research. But it's also, it's horrifying, and it causes, it's sort of a miracle that any drugs come out to be of any benefit to human beings based on what you've said today. It sounds very much like what it has the potential to make virtually all of the research happening today redundant or dubious. I'm wondering about the ethics of even releasing these drugs onto the marketplace. Is there oversight regarding that? Because these are such dubious results. Is there any kind of oversight in Canada regarding the release of drugs that are founded in this type of environment?
Yeah, there is, and that's why it's one reason why it's such an expensive problem. Because once say a therapeutic drug looks as though it's promising in animals, it's then moved to human clinical trials. So there might be additional animal checks for various safety issues. Then it's run through human clinical trials of different levels. I'm not an expert in this, but there's a number of different levels which become increasingly large and expensive. And that's where drug companies, I never thought I'd feel sorry for drug companies, but I actually feel sorry for drug companies. They are hemorrhaging money chasing up these false leads, because yes, you can't allow anything into the human population which either isn't going to work or is going to have some terrible side effect. Obviously, every now and then that happens anyway, but yes, these clinical trials are where they're trying to filter out things that would be inadequate or bad. They're done well, they're vast, they're extremely expensive, and that's where there's just an incredibly high attrition rate of promising drugs. Just nope, failed. Yeah. And actually, sorry, I meant to answer one of your questions. One solution is actually academics stop being allowed to do biomedical research, and it's all done by drug companies, because they, much as you may deeply distrust them, at least they're trying to find things that actually work. Whereas people like me, honestly, we're trying to publish papers and train students, and it's kind of different. It's not the same thing.
But I would think it would be the drug companies then who would want to see good conditions for the animals, because clearly those conditions, even if you accept animal testing, but those conditions do not equal good science. There's this profound waste in using the poor conditions. They should be at the vanguard of making those changes, because they're the ones who would save the most money.
Yeah, and some of them are actually. So there's a real move, especially for some of the European-based pharmaceutical companies, to just go beyond existing norms because they're recognizing, well, if the drugs are going to fail, they want the ethical price to be, the welfare costs paid by the animal to be smaller, and they want their drugs to be more likely to succeed. So yeah, pharmaceutical companies are very critical of the status quo, and they do want to make a difference. That said, that's probably the most forward thinking of them. There's still, many of them still farm out basic aspects of their research to companies in the developing world, so Thailand, and you farm out your work, farm out a lot of the work to labs in China where there isn't regulatory oversight and where welfare conditions are probably going to be terrible, and that's to save money, but I would say is probably a false economy. Aside from the ethical horrors, is a real false economy. So we'll see what happens there. Yeah.
Yeah, and I think also just jumping on that as well, the scientists in these companies were trained in universities. So there's sort of path dependencies about this is the right way to do it. And you get trained that if you create a complex environment for the animal, you're actually doing something wrong against so I think there's like the scientific culture even is feeding into the drug culture as well, and then you have these path dependencies. And we had a question up at the back, and then I'll also just jump on with this to say there's also the sister problem of not just drugs that we think work being tested and not end up working, all of these drugs that in theory could have worked that we miss. I mean, that's a huge lost cost and missed opportunity that I think also needs to be emphasized. So yes, please, in the back.
Thank you. First of all, thank you for the amazing presentation. I also believe that the more we can do to improve the welfare of these animals is important. I want to share that because of my work as an attorney in Spain, I have seen evidence of laboratories altering the outcomes of the animal testing for financial gain. So the science, this is my belief, the science part of this whole business I think is secondary for the profit. At least I saw that in one particular case. So since animals research is a commercial business, the primary incentive is often clients and to receive the laboratories more and more funding. So rather than science accuracy. And because of this reliability of animal research, that even if you improve welfare or if you follow the rules, nothing's going to guarantee that this is going to work in humans. I don't know if what are your thoughts that in this, like the main problem is actually the profit part of all of this?
I mean, I think you're right. It's a complex multifactorial problem. Animal welfare issues are only part of it. Others are, is it ever valid to extrapolate between species? And then there's all the issues around scientific rigor and relevance and external validity, which are open to unconscious or even deliberate biases along the way. So yes, I think it's a complex problem. I think for academics, where we're rewarded for gaining grants for which we need to publish papers. So the incentive for us is to manipulate data or analyze data until we find a statistically significant result, but it's not about making money. I think anyone would be a real fool to manipulate data to try and make money from a pharmaceutical product directly, because it's going to backfire if it doesn't actually work or if it's dangerous and you get embroiled in loads of lawsuits. So I think it'd be very short-sighted in the commercial world to manipulate data, because it's going to catch up with you and it's going to be expensive when it does. I do kind of think academia is more the problem, because we are in something of an ivory tower, and where what we're incentivized to produce, I keep saying the same thing, is not necessarily truth. And I hate having this message, because I don't want to foster conspiracy theories in a world where, truth is not necessarily respected and people don't believe in climate change because they don't want to believe scientists. Obviously, I think science is the best thing we have, but it's just very flawed and it needs to do a better job. Yeah.
Yeah, the science culture.
Yeah,
it's not
exactly.
actually following its own standards.
Its own ethos, yeah.
Well, you're an inspiration for how good it can be. So thank you so much again for your talk and for your work.
Well, thanks for the great questions.